We are live, ma'am. You can begin.
Good evening, ladies and gentlemen. Welcome to the second episode of the PCOS Science Live program. This is a program which is based on stimulating young minds to do research through interactive discussions and sessions with world-renowned researchers. This heart-to-heart discussion with them in the field of research will bring to you the most exciting origins of what we accept as science today. What occurred as research maybe 100 years ago is considered as part of clinical practice today. Each episode of these programs will bring to you how the idea was germinated, how it was researched, what was the inspiration, who was the mentor of the researcher, how many years did it take, how much resilience was there, and what did it finally lead to. This program is being held once in a month, and the last month when we had our first episode, we had the pleasure of having with us Emeritus Professor Praveen Matre. The world's first ovarian transplant was done by him. And if you have missed it, please check it on the PCOS Society YouTube channel. You'll be able to see the entire program. Even this program today will be put up on the YouTube channel of PCOS Science Live. Our second episode today is as exciting as the first one. And today we focus on COVID and human reproduction. COVID is the buzzword today, and this represents a unique global challenge for all of us, a pandemic which has really paralyzed the whole world. The timeline to complete resolution is still not known. When it will go, how it will go, we still don't know. But it is important for us to prevent the spread of infection to both our healthcare workers and to patients. And this is something which we have been doing by the constant, staying away, keeping distance, wearing your mask, and doing all what is necessary to prevent infection being spread. But at the same time, there is a lot of ongoing research in the country and all over the globe, which we hope that these new developments will bring us some respite and will give us some information on how we could overcome this challenge. So what we need to do today is learn that how does COVID affect human reproduction? We know that it affects the lungs. We know it affects the coagulation system. We know about quite a few of the organs, but there's not much known about human reproduction. We know that there are mothers who are COVID positive, who have delivered babies who are COVID positive. But how that is happening, why it is happening, this is where our research is going to go. So today we have put together the team of researchers from the Institute for Research in Reproduction, which is a part of ICMR. And this team is doing fantastic work at the NIRRH in Mumbai. And this institute celebrated its 50th anniversary this year. As per our agenda, we will be discussing first the paper which has been published by Dr. Deepak Modi, a researcher from the NIRRH. And Deepak will be talking about his paper which he has published, which is quite a complicated name actually. So what I'm going to tell you, what it talks about is how the human placenta transmits the virus from the mother to the fetus. And it has been published as late as 19th of August, 2020. Now, this is something which we will discuss from Dr. Deepak, what this complicated title of his means. And therefore, I would like to welcome Deepak on this program and talk to him about his paper, which has made great advances. So, Deepak, I would like to welcome you. And you are the corresponding author of the paper on COVID, which tells us about how COVID is transmitted vertically through the placenta from the affected mother to the child. Now, if I had to introduce Dr. Deepak, Dr. Deepak is, look at him and he looks so young. We don't know how old he is, but he's an accomplished scientist at the NIRRH. He has a doctorate in human genetics and is currently a scientist F at the ICMR-NIRRH and heads the Molecular and Cellular Biology Division of the whole group. Deepak has won numerous national awards, and he has also been listed here with his 85 publications in prestigious journals in his portfolio. And this is the right person with us today who will guide us on how research is done, how to take it to its logical conclusion. Currently, Dr. Deepak's area of interest are the understanding of endometrial receptivity and endometrial implantation, endometrial disorders, genetics of sexual development, and his latest love is COVID-19 and reproduction. So over to you, Deepak. Welcome. We are happy to see you here. And we can start the discussion on your paper, which has been published recently. So, as I was saying that Deepak looks very young. So, normally the saying goes that ask a woman her age or you ask a man his salary and you will hear no lies. So, I'll ask you how young you are and I'm sure I will hear no lies. So, how young are you?
Sure, definitely. No, so first of all, that's the beginning. That's a lovely beginning. So first of all, thank you for having me here, and it's an absolute pleasure. And thank you, PCOS Society, for selecting me and the Institute for this wonderful chat show. And I'm really very nervous because it's the first time ever that I'm doing a live chat show like this. So I'll have to think my age.
Come on.
Yeah, so I am 47, 1970.
47, very young. So he's a grade F senior scientist at NIRRH. And so what I want to ask you is, you obviously went to school, you went to college. When did you decide that you wanted to go into research?
Yes. So, the current notion would be that, everybody thinks that in Gujarati we say, ke, chokra na lakshan parnama dekha, like you see the features of the child in the cradle, but there was nothing like that in me. I was absolutely a very average student, didn't like studying much. I couldn't, mug up things and just go out, just giving out an exam. So I was a very average student in school, and I'd never decided that what I really want to become till I decided to graduate in zoology. That's after my second year BSc. And that's where I thought that, no, I really need to do something for my life, and it has to be something interesting rather than a mundane 9-to-5 job. That was something which I was fairly clear. So, and then because I liked biology since school, so I thought that, okay, let me major in biology and see. And you can imagine this is the time I did biology when it was supposed to be meant for girls. Girls who do not get married do biology. And here I'm a boy in a class of a lot of girls trying to major. So you can imagine the kind of situation that I was in. But because I thought I loved it, I just did it. So that's how my journey as a scientist began.
Was there anyone who sort of inspired you that you need to go into research, or was it just a feeling that, okay, I don't know what else to do, let me do research?
No, it was not. Yeah, so in a way it was like that, because till my second year BSc I was not clear what I really wanted to do, but it was my college teachers who really made us read through a lot of research papers, made us read through what is happening latest in the field, and that kind of enticed me that, okay, this is how people generate new knowledge, and that's where the interest started coming in. That's where I wanted to do something which would kind of make a difference to people, make a difference to understanding of science and biology. And that's where I thought, okay, let me finish up my graduation and then see. So it was more of a walking through the path rather than deciding a path.
Any challenges whilst you took up research?
So even before, when I decided to do masters in biology, people really call me stupid. Imagine coming from a orthodox middle class Gujarati family where most people either start working or get into business. I'm saying here, no, I want to do 2 more years of studies, and people said stupid, and then they said, oh, biology, more stupid, because it is a time of computer era. They said, hey, computer science karile, why do you want to do biology? I mean, I'm a kind of scope. The typical Gujarati family.
Right.
And then I said,
all
no,
the
not
Gujaratis
really.
will be mad with you by now, huh? Okay.
Yeah, I call myself a mutant Gujarati by now because I don't know how to make money. I know how to spend money. I think Dr. Mahale will agree with that I can spend more money than actually earn. So
okay, good, not bad. Anyway, so the other question I have, I read on your CV, you're scientist F. Now, what really does that mean?
Yes. So in different government systems. So once I finished my graduation and my PhD, and then I thought that, okay, the research is in this country is done in institutes and universities. So I thought I would want to do in an institute because that will give me more focused way of doing work and not having too many challenges of teaching. But I love teaching. So, but I joined this institute, and in a typical ICMR system, you have various categories of scientists. So, entry level is scientist B, and then every 3 years and 5 years, you kind of jump up. So, it's like B, C,
Okay.
D, E, F. So, it's about the fifth promotion that would get you to F,
Okay.
which is just below the senior most position.
Okay, all right. So, you're almost there.
Yes.
Okay, so how did you decide to join NIRRH? Were you somewhere else before, or you straight away joined there?
No, so after completing my graduation and post-graduation Wilson College in human physiology, I was not sure whether I wanted to teach or I wanted to get into research. So I taught there for 2 years, but I realized that I cannot teach all alone. I mean, I'll need some spice, and spice came from doing science. So then I joined the Wadia Hospital for Children, which was a vibrant place at that time, which was offering a lot of research opportunities. And that's where I started doing a PhD in human genetics. And this kind of set the stage that, okay, I want to do something. And where I would see a lot of women, I'm sure you were there when I did my PhD, used to visit the maternity hospital that time.
I was a visiting consultant there at the Wadia maternity.
Yes. I do remember seeing.
I was very much at NIRRH. I used to come there to the library to read, and I used to have a lot of friends there. So it was very good for me to be there at Wadia. I spent a lot of time there. Right. I was coming to the part where we would like to discuss your paper. And I'm sure you've had so many papers before, but since this is current, this is very new, and this is something which I'm sure most of the gynecologists are interested in. So what I want to know is that, what got you interested in COVID?
The COVID pandemic, I mean, it started as a very phasey, fuzzy situation where we weren't sure what's happening, whether even we'll get it in India or not. It was, I remember very distinctly, I was coming back from the ESAR meeting, which happened in February end, and I realized that COVID is a serious business and many people are infected. So it's most likely that the IVF fraternity would also be facing these problems. And while coming back in the flight, I was thinking that how my expertise in placental biology, early embryonic development, I can help in understanding COVID. And that's where I thought that, okay, let me look at the coronavirus receptors. Are the reproductive tissues, are the embryos, are the placenta having these receptors? And that's where I came back and I told my students that, look, this is going to be a serious problem. The COVID is really getting into the business, and let's see if these tissues, or as reproductive biologists, I think we should contribute to it. And that's how I made the beginning. And it was a very, very abrupt thought of, trying to do something very out of the box.
So you got an idea, and then how did you proceed? Now, this is meant for our young students who want to understand, okay, I have an idea. What do I do with it? How do I go about it?
So in the research process, our idea then sets into a plan. So once we have an idea, usually I see a lot of people jumping to really answer the question that's coming in the idea, but that's not the way one has to do. One has to then sit and make a plan. So what we did was we sat in the lab and said, okay, what is the kind of research question we want to ask? But okay, now the bigger question was, does COVID affect reproductive tissues? Does it affect the embryos? Does it affect the baby? Does it affect the pregnant women? So now how do we address it? There are many, many ways of doing it. But then I had to look, what are my strengths? What are my weaknesses? What are the opportunities I have about? So when coming back, we realized that our strength is looking at all the available data which is there out. So we made a plan that, okay, let's systematically work out what are the known receptors for COVID. Now, the problem that we started facing was at that time, we didn't even know how COVID entered inside the cell. So my students had made a plan that, okay, this is how we will start figuring out what receptors are there, known receptors. Then we still say we'll start looking at in reproductive tissues. Where are they expressed? Then we'll start looking at what all they are doing in those tissues. So it was like a plan which we made over a period of time, and then the lockdown set in. So then we all had to go to our home. So we really couldn't do any experimental work. And that's where we decided that we will do all the work, which is the data available with us, where we have access to it, and we try to address that question.
Mm-hmm. Okay. So, but you have done work in your paper on receptors, etc., which I would want you to talk about. But basically, I wanted to understand the process of how does one go about this kind of research when our students have an idea. So you said you get an idea, you make a plan, how you'll go about it, go with what your strengths are, and then work in that area. Correct?
Yes.
But as clinicians, not many have the opportunity to do that research, which in an institution you could do. Similarly, in a teaching hospital, even though it's a teaching hospital, it's not easy to do any research because we don't have the funds to do it, etc., etc. So,
Yes.
so how do you go about it? Do you have to submit a proposal? Do you have to first get funding? How does it work?
So those are a part of the plan. I mean, I would at this point disagree with you. I'm sorry for that, and for most people, that I don't think clinicians don't do research. I love
I
this
don't agree.
quote from I love this quote from Albert, which says that discovery is all about seeing what everyone else has seen, but thinking what no one else has thought. And as clinicians, you do it every single day because every patient that you see is not the same which you just saw 10 minutes back. And every patient, you see it in a different way. You think in a different way what the patient is going through. And medicine, as you know, Siddharth Mukherjee has said that it's all about telling stories. The patient tells the stories to tell about the illness. The clinicians tell the stories to talk about, they describe the illness. The clinicians describe the illness to understand it. And that's what science also does. It's all about telling stories. So I don't think we should really inhibit ourselves thinking that, research is something very different from what the clinical practice is. Every single day you see new patients, and that's new science. It's just about looking at it in a very different way. And that requires some amount of thought process, some amount of thinking, which I think everybody has. It's just that you have to just change the frame of mind and get it there. So that's what I would say. In a practical sense, yes, once the plan is made, we need to figure out what are the things that we would require. And if, of course, a lot of research does require money, so you have to figure out that, okay, if this is going to require X amount of money, where I'm going to get from. And that's where funding comes in picture. That's where ethics committees come in picture. But those are the very small part of the plan. The beginning starts with a research question. What is my question and how am I going to approach it? And once that's done, 90% of the job is done. It's just then putting the pieces of the puzzle together.
Okay, all right. So if you tell me, when you thought of your plan and you published your paper, can you briefly describe what really did you do? How did you go about doing it? And what did you find at the end of it?
Sure.
I know you're a research scientist working in a lab, so you have terminology sometimes which clinicians don't understand. Just as I don't understand the business words completely, similarly, you may not understand the clinical terms we use. Like say for sometimes, some people, they write at the end of the notes, the houseman used to write, GOK, diagnosis GOK. What is GOK? Nobody knew. Till we got to know, God only knows. God only knows. So its question is, what really we want to understand what really you did and what this paper is about.
Sure. So in fact, we had two papers. One is under process of review, but which is in the public domain, which we now call it as archives. And the other one is published in Frontiers in Developmental and Cell and Developmental Biology. So one is on the developing human embryo. So the question that we asked was that if any woman who is undergoing IVF in a clinic, and if by any chance that there's an exposure of the virus to the embryos, can the embryos get infected or they are spared? Our idea was that if they are spared, then it's safe to do IVF during the pandemic. We need not worry about it. So the way our ideal way would be to actually take human embryos infected with the virus and see if it's really going in. That would be the most logical way of doing it. But obviously, you can understand that the virus is not available. We don't have the necessary infrastructure to grow the virus. Nobody would give human embryos to infect and see this.
Absolutely.
So we thought that, okay, let's make it more simpler. Let's break it down. So what we do is break down science. So we broke down here and we said that, okay, let us see if the receptors for the virus are present onto the cells of the embryo. Now, how do we know that the receptors are present? Okay, one way of going about it is take again the embryos, put the antibodies, and see if the antibodies show the presence of receptors. But that's not what we thought we would want to do. Let's see, because in the last 5 years, a lot of interesting work has happened where people have done something called as single cell RNA-seq of the developing human embryos. What they have done is they've taken IVF embryos, they've disaggregated each of the cell, and each cell they've looked at the RNA profiles of it. Now, the good news here is that all this RNA profiles is there in the database. I'll remind you that all RNA makes proteins and proteins will be the receptors. So we first thought, okay, let's look at if ACE2 receptors are there in the developing human embryo. So we had data from zygote, eggs, early zygotes. Then we had data of the 8-cell embryos, and we had data of the blastocysts and the developing blastocysts. So we tried to see if ACE2 receptors are there, and we did find them. So we had to do a lot of informatics here, and I'm thankful to my students who really worked remotely to do this work. And we found that the receptors are there, the RNA for these receptors are there on the inside the embryos, right from 4-cell stage. So as the embryo is developing, the ACE2 receptors start coming from 4-cell stage. They come up to the morula stage, and the levels go down later. So that told us that yes, the early embryos have the ACE2 receptors. Now, the virus doesn't enter just by using ACE2 on the cell surface, but the S protein of the virus has to get cut. And that requires another enzyme called as TMPRSS2. So we asked whether now if ACE2 is there and TMPRSS2 is there, and only if TMPRSS2 will cut the spike protein here, that the virus can enter inside the cells. So we found that TMPRSS2, for good news was that the TMPRSS2 was not present in the 4-cell stage, neither it was present in the developing blastocysts. So we were happy that, okay, the blastocysts don't seem to be at a higher risk of getting the infection. However, let's remember that there is still a small amount of this present. Now we submitted this paper, and the reviewers came back and said that you haven't looked at the trophectoderm, which is going to be the most exposed part to the virus. So we said, oh shit, how did we miss that out? So we make up these mistakes. We often make this mistake. So we send off, and the paper comes back, it gets rejected. And then we started looking at this data all over again. And now we started looking at trophectoderm. And that was a disaster. We found tons and tons of ACE2 present on the trophectoderm, right from the blastocyst hatching stage up to the late blastocyst stage. So this told us that along with ACE2, there was TMPRSS2 there. Then at that time, when we were analyzing this data, new receptors for the virus were discovered, what we call it as CD147 or basogen. And we found that even these were present on the trophectoderm as well as some cells of the inner cell mass. So this is the first evidence that we showed that the human embryos, developing human embryos, are permissive to the coronavirus, and that set the stage. And then we repeated the same story with the human placenta.
Is this the first time it's been reported in embryos?
Yes. So when we put it in archives, it is the first time that it was published in human embryos. After this, 2 papers have come which have shown the same thing.
Okay. So do you get the credit for being the first or no?
Yes, I do.
You do?
Yes, we do.
Wonderful.
So that's the advantage of putting in archives. So although it's not peer-reviewed and still published in a full-fledged journal, it's still under review. So you realize it's a very longish process. But even before the data is ready, we can put it in public domain and tell the people, look, here it is. You need to be careful.
Okay. So how does one do that? How do you put it in the public domain?
So in the public domain for medicine, there is medRxiv, there is bioRxiv, and there is archive. And now, of course, there's preprints in number of servers. So all of these journals have the preprint servers. What you can simply do is prepare your article, put all the figures, put all the right references, and make a PDF and just upload it there. In about 24 to 48 hours, it just comes online. And it has a DOI, which can be shared with everyone.
Okay. So and then in terms of that you do get credit for it, even though it's under review. Am I right? Perfect. Yes. That's brilliant. So you found that the embryos were, the embryos were showing evidence of infection, even in the trophectoderm, and was it also infecting the inner cell mass of the embryos?
So the inner cell mass doesn't contain much of the ACE2 receptors. It doesn't have much of TMPRSS2, but inner cell mass does have the alternate receptor, which is basogen. So it is likely that the whole embryo is most likely to get infected.
Most likely to be infected. But why did you think of looking for ACE receptors?
So ACE2, Yes. So in the lung, the ACE2 is the receptor through which the virus infects the lung. So that's an entry point in the human body.
Okay.
So we thought that the same receptor should be used by each and every cell, and that was the logic which we applied here.
How does one find out which receptor to look for? There are hundreds and thousands of receptors.
Yes. So
So
that's
even the people who did the research on the lungs, how did they think of ACE receptors?
So this is usually historical, and so we know that coronaviruses is a large family of viruses, and humans are known to infected by it. So when the first SARS infection was reported way back in the 90s, that time people were doing hardcore biochemistry experiments. So they took cells and they put the virus and they see to which protein it bound, and then reported it was ACE2. Now this coronavirus and that coronavirus is a very close sequence similarity. So it was
Okay.
first a suspicion that ACE2 would be the receptor, and then other researchers actually did experiments to show that if you remove ACE2, the virus cannot enter in. If you give extra ACE2 to cells where it's not supposed to be there, then the virus enters in. So there are set ways of looking at it. And
Okay.
once that's proven, then other researchers take over from this. As you
Take
would realize that research is not single man's effort. It's a community
Absolutely.
effort.
Goes over years and years and years. Yes, we
Yes,
know
and that's
that.
where a lot of things take a lot of time.
So the other question I have with you is that if we have to talk about this virus, as you said, it's a group of SARS viruses, and each one is a little different from the other. Uh, we call it the COVID 19 virus. Probably it's come in the year 2019,
Yes.
right? But
Yes.
why is it called novel? I've not understood that.
Huh. So, because it's new, so as I said, that the coronaviruses, there are lots and lots of coronaviruses out there, but
Hmm.
this and many of them infect humans, many of them don't infect humans. So till now, we knew that SARS was one which caused the respiratory disease. Then came in MERS, the Middle Eastern coronavirus disease, which
Yes.
came about some years
Yes.
ago. And
Yeah.
this virus did not have any sequence similarity to those two.
Okay.
So the coin novel was added to it, that this is a new virus which is found. So in terms of evolution from the first family of coronaviruses, these viruses undergo series of mutations. Some of the mutations that happen, the changes in the DNA sequence that happen, are the ones which cannot make it infective to human. They don't bind to human ACE2. They may bind to monkey ACE2, or they may bind to some other ACE2s. But the mutations which occur in the spike protein, which allows binding to humans, this is a new virus. So that's how the word novel coronavirus came
All
into.
right. But
So
this
in
is the
fact,
only one. But there have been other viruses which have been infecting the human beings, and they are also coronaviruses. This one
yes,
is the background information is it's coming from a bat. Is that
yes.
true?
Yes, so the,
It's
the
coming from
sequence
a
that
bat.
it's coming from a bat. So something happened in the bat that when these viruses are very notorious in terms of that when they undergo replication, they undergo division. There are a lot of DNA level changes, which I mean the nucleic acid level changes which
Yeah.
happen there. And because the
Yeah.
machinery is not very nice, it makes errors. And in this errors, it lands up forming a new virus.
Okay. So
So
after this paper which you had on the embryo, there has been case reports, and there have been reports on in pregnancy where you're looking at transmission through the placenta. That
yes.
paper of yours which is showing that there is a transmission through the placenta to the fetus, I think it's important to discuss that, because sometimes when babies are infected at birth and the mothers are infected, we don't know whether the baby got it from the mother because the baby is feeding with the mother and in close proximity to the mother, or is it that it was an in utero transmission to the baby. So what was the research you did to determine that?
So this was a precise question which we had, that there were reports of mother baby diets being positive, and this was and the belief was that the babies got it after birth through extrinsic, or what we call it as a lateral transmission, not a mother to child transmission. So we said that if there is to be any evidence of mother to child transmission, remember the placenta is the barrier which prevents things from the mother coming into the baby. So to address that, we thought that if the placenta is protective, it should not have any receptors for the virus. But if it is not protective, then it should have the receptors for the virus, and the virus has to come here. So again, replicating our embryo model, we first took single cell RNA-seq data of the first trimester placenta because that was what was available in the public domain. And we tried to see that are the receptors there. And yes, that was we did see a whole lot of receptors in the syncytial trophoblast. So the placental villi cells have the receptors, the trophoblast cells have the receptors, the fetal endothelial cells there have the receptors. So we realized that the placenta does have the receptors for the virus. There we went even one step ahead and tried to see that if the virus infects the placental cells, then can it replicate inside? Now, for the virus to replicate inside the cells, it needs to have the necessary machinery because it uses our genes to replicate. So, are those genes present in the placental cells? When I say genes, I mean RNA. And are these RNA present in the placental cells? And yes, we found that the placental cells, in fact, have a very high abundance of these genes, which were required for viral replication. So we first showed that the human placenta, at least in the first trimester and the second trimester, is what we call it as permissive to viral infection. That means it can permit the viral entry, and even it can allow its replication. This was where the beginning to generate evidence that yes, the mother to child transmission is fairly possible. In if the mother is infected, we would get the
But you
child.
think it's more in the first trimester, mid trimester, or in the last trimester? What is the
Again,
current evidence?
yes, so that was one question which again the reviewers asked us. So, we as researchers have a favorite question and we answer it and we are happy. But the reviewers are the ones who keep us on our toes and tell us that no, you have not shown this, this, this. So the reviewers did ask us the same question, and that was a challenging thing because a lot of data was not available. But thanks to again my smart students, what they did was they took instead of taking single cell RNA-seq, they took the total RNA-seq data of not just human placenta, but they took placental data from about 7 to 8 species, which is now at term, third trimester at term. And in all the species, we did find the ACE2 receptors and the TMPRSS2. So this told us that throughout gestation, the placenta does express it. So now if the mother gets a virus, it gets to the baby. So this is what we found. And then eventually we found a very interesting case report, which I would like to discuss once we go ahead in this. So you would want me to tell about it right now? Would that be interesting?
Okay, we'll take it a little later.
Sure.
At this point in time, I would like to bring in Dr. Smita Mahale, who's the director of the institute, and she is the person who's heading this whole organization where such amazing research happens. And I'd like to invite Dr. Smita Mahale, our special guest for today, the director of NIRRH. Welcome, Dr. Smita. It's so lovely to see you. Such a heartwarming person and such a brilliant smile. I am very, very happy to introduce her. And she's a very warm and unassuming lady with no airs about her, even though she has occupied the highest position at the NIRRH as its director. Dr. Smita is a current grade G scientist, that's right at the top, having joined NIRRH 50 years ago. And you won't believe it, she had the honor of celebrating the 50th anniversary of NIRRH recently this year, where the minister, the health minister, was there as the chief guest. And it was a wonderful program there, and I was there, part of the faculty, and I was really enjoyed that program. Dr. Smita's research experience spans across 35 years in reproductive health. She has over 80 peer-reviewed articles in very esteemed journals, and her main area of focus had been gonadotrophins and their receptors. As gynecologists, we use so much of gonadotrophins today. We use the antagonist, we use the agonist, and I know the exciting time when the receptors were first discovered, and it was an amazing experience to hear all the researchers talking about it. She has over 85 publications in prestigious and national journals, and she has various awards and honors. Now, the awards and honors are plenty, but the most important ones are she's a fellow of the National Academy of Science, which she was conferred in 2010, fellow of the Indian National Science Academy in 2015, the ICMR Award 3 years, 2003, 2012, and 2017, and the Royan International Research Award of 2008. So, Dr. Smita is an extremely knowledgeable person, and I know her personally because we have collaborated together on the PCOS Abhiyan, which is a collaboration between the Institute, the PCOS Society of India, the Tata Institute of Social Sciences, and the Kasturba Health Society, headed by Dr. Rama Vaidya. And the sole purpose of this is to bring the relevant disciplines together so that we can work towards the cause of PCOS. So I welcome you, Dr. Smita, on this program of COVID and human reproduction. So, Dr. Smita, your institution has been doing a lot of work with COVID. I've seen the papers which have been published. We heard our Deepak Modi talk about his papers. And we have also heard about the various areas NIRRH is working on with COVID. I know that during the lockdown and during this whole period, besides doing your research, you'll have served as a service organization for taking care of COVID patients. And I think you'll have done a tremendous job. And I would love to hear you say what you would like to say about this COVID and human reproduction and your institution.
Thank you, Dr. Duru Shah. It's a real pleasure to be as part of today's webinar. And as part of ICMR Institute, we had to shoulder a lot of responsibilities. And most of the viewers would agree that ICMR has taken a major role in the government programs. So when there was a call, I would say the response from the institute was expected by ICMR and also by the ministry. The response expected was at the local level, at the state level, as well as national level. So we all know that Mumbai was the main first city which was affected. So we got a call from Municipal Corporation of Mumbai, and they said, "You have research facilities. Can you help us?" And with the existing small virology facility, we thought what best we can do. We identified some of our research scientists, whom we call them as Corona warriors, and two of them are there in today's program, and couple of others were part of this whole exercise. So to start with, we had this very small laboratory as per ICMR guidelines. And as and when there was a need, there was again responsibilities taken up. And ICMR also helped us to establish a high throughput COVID diagnostic laboratory. And that was inaugurated by Honorable Prime Minister during the month of July. So that's how gradually we increased our capacity of testing, and at present we can take up almost 800 to 900 samples per day. So the samples come from all over Mumbai, neighboring districts also, and that's how we started this testing facility. But along with this, we had to take up other responsibilities because the central government was supplying the reagents. So we were made part of the depot for supply of reagents. And it was a major responsibility because we had never done it in our entire career. And it had to be done in a professional way. So all the labs in Mumbai, they were supplying the reagents, of course, the government labs. And then slowly we were given the responsibility as a zonal depot where we had to give for the Western India. So even the neighboring state. So that's how we started. And while doing this, we got into the validation of the kits, the quality assurance of the kit, and also as part of the Atmanirbhar Bharat program of Government of India, we were told to handhold these Indian companies. So they could approach us with their reagents, and we help them in validating, giving the reports. And that's how you can see at present the costing has come down a lot, because there are competitors. The Indian companies came into the market. There was a lot of competition. The quality assurance was done at each time, and we are in a shape now. And if you look at the last 7 8 months, it started with one lab in Pune, NIV, our sister institute, and today ICMR has almost more than 2,000 labs across India. So as and when there was a need, we did pitch in. We provided training to the people, and we also, as part of our institute activity with the state government, it is a program of Model Rural Health Research Unit. We have one in Dahanu, Palghar district. So we could set up a RT-PCR lab even in that area way back in May. So that was
Right.
first of the rural labs doing RT-PCR, and again it was a collaboration with the district health officers and the collector. So that's in short the kind of the testing facility or the diagnostic facility we offer to the government and to the local government.
Hmm. So, besides the research, you are also providing support in terms of diagnostics and testing.
Yes, yes,
But if you had to talk about the kind of confusion there is about testing today, like say for
yes,
example, yes, we believe the gold standard is RT-PCR, right?
yes,
But
yes.
there are so many antigen tests coming into the market. There are so many antibody tests coming into the market. There are so many queries on how long does the RT-PCR remain positive after being infected. There are situations where people are admitted in the hospitals, and then they are kept for 7 days, and even they are not tested before discharge after 7 days because they are totally asymptomatic, and then they are told that no, now you can go home. In short, there is a lot of confusion about this information about what to use, what test, and how long does it remain positive. So can you throw some light on that?
Sure, sure. So the strategy of testing
Hmm.
kept on changing because
Yes.
this was a new disease and
Correct.
nobody knew how to manage it. And when we started, we didn't even have the capacity.
Yes.
So individually, one could not go and get themselves tested. So there were
Correct.
some guidelines issued. So the initial targets were like symptomatic people or people having severe disease problem like SARI. So that's how we started. But the gold standard still remains a real-time RT-PCR test. Eventually, when the other tests started coming, the purpose is different because we know real-time PCR takes 4 hours, 5 hours. The turnaround time for a public health system, it is almost 24 hours. So the rapid antigen test came into market. So the difference here is in the real-time PCR, we are looking for the RNA of the virus. So when we see the rapid antigen test, we are looking at the protein present in the sample. So that's how we went ahead. And for the rapid antigen test, there are issues because the sensitivity and specificity, there are couple of problems there. But think of a situation, a patient comes to a hospital with severe clinical symptoms, and there is hardly any time to get the RT-PCR test done.
Correct.
So
Correct.
in that case, these rapid antigen tests are very, very handy.
Correct.
And if the results are positive, that means it
Yes.
is confirmed positive.
Correct.
The challenge there is if the test is negative, one should not
Yeah.
just leave it aside.
Correct.
So if it is negative, we would say whether it's in spite of whether they are symptomatic, asymptomatic, better to refer them for RT-PCR tests so that at least you rule out this
Yeah.
infection. So
Hmm.
that is one part. The rapid antibody test has its own advantages and disadvantages. There is a lot of confusion in the mind of general public. I would like to stress here that rapid antibody test is not a diagnostic test. Okay? It is meant for looking into the community. How many people were affected in the past? It's kind of a zero surveillance, but it does give information like we read in papers, like in Dharavi, like it was around 50% people were affected. When whether whereas in high-rise buildings it was only 4 or 5%, and that is showing up now. So now, if
Hmm.
you look at that incident, so it gives some kind of a hint to the public health officials where to invest or where to target. Because now Dharavi and the slums, it has come down because almost 50-60% of the people
Already.
they got the infection, and whether it is asymptomatic, symptomatic, they have antibodies now.
Correct.
And now, as we see, the incidences are going up in the localities where we have these housing societies or
Yes.
these high-rise buildings. So
Yes.
that is, this is useful in a different way. So it is up to us how best we can make use of these tests and interpret the results.
Mm-hmm. Okay. So, is there anything more you would like to tell us about the institute in terms of what else you are doing with research, or is there any new technology you're using for anything?
Yeah, there is a lot to say, but I think because of time, I wouldn't go in depth. Uh, when the COVID, all these, like we were doing testing and report work, we all sat together and discussed. I said we must do some continue doing research,
Yes.
and we planned research in the area of COVID itself. So the one part of the COVID registry in pregnancy, my colleague Rahul will take up. So
Yes.
other part, what we are doing is looking at the immune signature. It's a basic study where we are looking at how individuals behave when they get infected, what kind of antibodies are generated, and can we predict the progression of the disease? Which person, looking at the kind of antibodies or the interleukin levels, how will they progress? Whether they will become severe or they will be able to cope up with the disease. So that kind of experiments are going on. And for this, I must thank all our collaborators because we have a good networking and Kasturba Hospital, Nair Hospital, because all the samples were referred from them, and we do have this as a project which is going on, and we will be doing it in a collaborative manner with all the municipal hospitals in Mumbai. Apart from this, we were also part of the uh, actually the repository. ICMR had a call again and identified various institutes for setting up repositories. So all these samples,
Hmm.
it's a properly planned project for 6 months.
Hmm.
With the samples will be stored, and these samples will be made available for the researchers in future because the time is short and everybody is busy providing services, hardly any time to sit and plan a proper
Correct.
research.
Correct.
So,
Yeah.
so we have this biorepository which has been started, and again we have collaborators across Mumbai. And
Yeah.
apart from ICMR, other government institutes also have been identified for this. So all these research activities are supported by our parent organization, ICMR, and that's how we are going ahead with this. And one
Okay.
thing I would like to tell the viewers, I think the minute they think about research, there is some kind of a fear. Maybe it is not their cup of tea, but I would tell them clinicians are the best people who can initiate research, look for collaborators, look for people having technical know-how, develop some kind of a linkages. Institutes like ours, they are open for everybody. It's more of a academic collaboration. So you have to approach us through your parent college or institution. So definitely it is kind of a give and take relation, and one can do it in a systematic way. There are a lot of opportunities for youngsters right from their MBBS days. So they can write a research proposal. ICMR promotes summer research fellows. The final year MBBS students can write proposals, compete, get the grant. Even during their MD, there are some avenues where they can. And especially for medical graduates, there are special programs, and one has to avail these opportunities. And ICMR, for that matter, lot of human resource development for medical graduates and even the teachers. There are couple of fellowships available. So if you visit ICMR or DHR website, I'm sure you will get into. Even today, there is a call from DHR. The last date is 16th of November. One can write proposals and make best use of this facility. Thank you very
Thank
much.
you so much. Thank you so much, Dr. Smita, for that. I think it is something like a godsend, and I think all our young people and clinicians interested in research should take advantage of this. I'll come back to you later, Dr.
Yeah,
Smita. We
thank
will
you.
have a little panel discussion at the end of the session for half an hour. So, any questions which you have, please post them in the box provided, and we'll take up the questions with all our panelists. I would now like to invite Dr. Rahul Gajbhiye, who is another senior scientist at the National Institute of Research in Reproduction. Welcome, Dr. Rahul. And he's currently working there in the NIRRH, which is part of ICMR. Dr. Rahul has been a research scientist D at the institute, and he, after obtaining his PhD from the University of Mumbai, he completed his postdoctoral fellowship in reproductive genetics under the mentorship of Professor Montgomery at the Institute of Molecular Biosciences, the University of Queensland in Australia. He now is a visiting faculty there at the same university, and he is also currently the Frontiers in Reproduction Fellow at the Marine Biology Lab. He is the research advisor to the Endometriosis Society of India, being very much involved in the research of endometriosis. He is also the international ambassador for the World Endometriosis Society, and very much in that area. He has had many alliances he's created. He's like a reach out person. He reaches out to different things, and he carries the research with him, and he collaborates with people. So he's a collaborator with various other organizations like the Wellcome Trust, DBT, and Indian Alliance Fellowship. He has the Burroughs Wellcome Fellowship. So through all these fellowships, he connects with all of them and does a lot of collaborative research. But in the public health space, again, he's outreaching, but you'll be surprised to know that he's the one who is currently providing information and training on treating snake venom poisoning. I think that's a fantastic thing to do, which is so prevalent in the rural areas. And in short, he has also acting with the national research programs at 31 field units of ICMR located all over India. So in short, he is actively contributing to the public health policy development, especially disorders of public health importance at national and international level. And he is currently the principal investigator of the National Registry for Pregnant Women with COVID-19, which has been initiated by the Institute. And this registry is called the Pre-COVID Registry. So I'd like to invite Dr. Rahul to come and speak to us about this registry and tell us what really it is, why it has been set up, what exactly purpose does it serve, etc., etc. So go ahead, Dr. Rahul. Welcome.
Good evening, ma'am. Thank you very much for the introduction, and I would like to thank the PCOS Society as well as you personally for inviting me here to talk about the pre-COVID registry in this important webinar. We know that in the previous outbreaks of the influenza in 1918, there were many deaths and there were poor outcomes among the pregnant women as compared to the non-pregnant women. And when the COVID-19 pandemic started in our country, at that time, also at international level, there was very little bit information available with respect to impact of the COVID-19 on the pregnant women and their newborns. So there was a need to take up the national level policy research, and under the leadership of Dr. Smita Mahale, we launched this national registry of pregnant women with COVID-19. We call it as a pre-COVID registry, and this was launched in May 2020 in joint collaboration with the Medical Education and Research Department, Government of Maharashtra, and with the support of the BYL-NIRRH Hospital, which is a dedicated COVID-19 hospital. So this pre-COVID registry is a registry of pregnant and postpartum women with confirmed diagnosis of COVID-19. And through this registry, we are collecting the information on the social demographic aspects, the epidemiological aspects, the clinical presentations, the treatment outcomes, the pregnancy outcome, and the neonatal outcomes in women with COVID-19. And through this registry, we are also trying to understand the impact of the COVID-19 on mother and child together in Indian setting. And also we are trying to understand whether there is a mother-to-child transmission of SARS-CoV-2. Now, through this registry data, we think that we will be able to generate some evidence on policy issues. And this registry, we are hoping to run for at least 2 years, and if required, we may extend further. So from this registry data, we expect that the information will be generated which will be useful to the clinicians to improve the management of COVID-19 in pregnancy, and also to reduce the maternal mortality and morbidity which is associated with COVID-19. We also feel that at the end of the 2 years, the data will be useful to the policymakers, or also in between when we do the interim analysis, the data will be useful to the policymakers for formulating the guidelines to address the burden of COVID-19 in India.
Okay. But you said it will be only for 2 years. Are you not interested in knowing what's happening to the babies who are affected long term?
Yeah. So when we launched this registry, our primary aim was to generate the evidence on impact of COVID-19 in the pregnant women. Eventually, we may think of having a long-term registry, long-term cohort, and follow these women. But that will be we'll see how the pandemic goes and how the situation is there in our country.
But the other thing is I wanted to know is that are you all only taking collaborators from the public hospitals or government hospitals or even private practitioners? Suppose I have a patient who's pregnant and she's COVID positive, and I want to put her on your registry as an entry point for a patient who's affected, to know what's going on with her. So then in that case, could one do it as a private practitioner?
Yes, definitely. Currently, the registry is open to all public and the private hospitals. In the phase 1, when we started the registry, we had 18 government medical colleges under Medical Education and Research Department and the BYL-NIRRH Hospital. But we are open to join the hands with the private hospitals who are providing services to the pregnant women with COVID-19. And the detailed information on how to join the registry is available on our pre-COVID website. There are mainly 2 requirements. One is the COVID-19 hospital or the collaborator interested to join the registry will have to sign the memorandum of agreement with the NIRRH and also need to obtain the ethics committee approval.
Okay. So if you're not with a hospital and you're running your own private practice, the patient goes, say, she's in your own nursing home and she's delivering, would you accept those patients as on your registry?
Yes, definitely, madam, provided the nursing home is part of our pre-COVID registry network.
All right, I get you. So currently you have how many collaborators on your registry?
Ma'am, total we have 60 collaborators from the 19 study sites. We have a dedicated obstetrics software which captures the data of mother with COVID-19, and we have a neonatal data which captures the data of newborn delivered to the COVID-19 mother. So we have the collaborations in the OBGYN department as well as the pediatrics department. So we have total more than 60 collaborators at the moment.
Wow. And how many patients have you already got registered on the registry?
Currently, I checked the pre-COVID registry portal, and we have more than 3,200 women which are registered on this pre-COVID registry portal. And out of that, around 1,800 women have been delivered, and we have a mother-to-child diet available for around 1,500 diets we have available through this registry.
Okay, so you have quite a bit collected already.
Yeah.
So I think that's fantastic news. I think, keeping in mind what Deepak was talking about, effect on the embryos, right, where COVID could be also present in embryos. Are you including the IVF specialist into this?
When we started the registry, we had developed the electronic patient record form, which captures the information on whether whether it was a natural conception or whether it was an ART conception. So all the data collection software is already available.
Okay.
The question of involving the ART specialist, we are open, and it's a good suggestion that if the IVF community is interested in partnering with this pre-COVID registry, we would be happy to provide support and collaborate with them. And as I already mentioned, we have the formalities to be completed to become the part of the pre-COVID registry.
So we'll definitely be in touch with you for that. As a matter of fact, if you look at the international organizations like ASRM and ESHRE, and they have already included registries for the IVF specialists, because I think it's important that the infection should not start. Like, say, if there's a health worker who's handling the embryos, because today all patients get their COVID test done before coming into the IVF program. But if the health worker who's attending to those embryos or those gametes, what are the chances that they could transmit it? They're working very closely with the gametes, and if they are asymptomatic carriers, they could be passing it on to the gametes or the embryos. I don't know if it's possible, not possible, but theoretically, logically, it sounds it could happen. So I think it's something we need to seriously look at, and probably guide our patients accordingly and guide the health workers accordingly. And I think it would be a good idea if that could get included.
Sure. Yeah, that's a good suggestion. Yeah.
It would really be helpful.
Yeah, we can add that to the ongoing pre-COVID registry.
Absolutely. So thank you very much, Rahul. And I think we'll bring in all the speakers together and we'll start the last session of panel discussion, which is more the clinical implications of whatever research has happened today. So in short, I want to bring out, like, whatever work has been done, how does it affect the clinician? How does it affect the patient? How does it affect us? Because finally, whatever research is done in labs, it finally boils down to understanding the disease better and managing the patient better. I think that's where we come from. New inventions, new discoveries, new challenges, all that comes in. And I think this is something which we are really interested in. So, Deepak, I'd like to start with you. I hope you all don't mind me just calling you Rahul and Deepak. Dr. Smita, I'm going to call Dr. Smita.
No issues, you can call me Smita.
So, when it comes to, the embryos, you said that the amount of infection which can occur in a day 3 embryo, a day 5 embryo, or a first trimester pregnancy or a mid-trimester pregnancy may be different. So suppose when I run my IVF center and I'm doing IVF, would it be safe to say that if I transfer day 3 embryos, it would be safer for me to transfer them in a non-infected patient as compared to putting in a day 5 embryo? What would you say?
So as you rightly pointed out that because there are a lot of asymptomatic carriers here, the likelihood that the one who is handling the gametes and the embryos may inadvertently transmit it. And remember, the virus is quite long-lived on even the plastic and the glass and the metal surfaces. So in a lab, there's a good chance because again, these air-conditioned spaces. Because the receptors are low by day 3, and even because this embryo is covered by a thick zona pellucida, the chance of virus infecting inside is quite low. So it's nicer that we would change our practice at least for some time to at least transfer day 3 embryos, or at least freeze them by day 3, not get up
Exactly.
to the blastocyst till we are set
Yeah.
with the whole procedures.
Exactly. So we normally, when we have many embryos, we tend to freeze them at the blastocyst stage. And instead of freezing them at the blastocyst stage, if at all we freeze them on day 3, it would be a better idea, and not transfer them now till, sort of the pandemic is more under control or we have more information. But something, there are patients actually. I was talking to someone in the West, and they said, a good friend of mine, he says that actually there are more women coming now for IVF because they have the time in the world to come, number one. Second thing is they are, working from home, so they don't have to take leave at a particular time. They can just drop in the clinic, get their job done, go back home and work. So they find that the work is much more. Whilst at least in our part of the world, at least till now, women were not happy to come and get treatment done. And frankly, many of us had kept our IVF centers shut because I felt that it's not right to do the IVF at this stage. But this is a very important clinical point which we must take home, that ideally don't do it now. But if you have to do it, then freeze the embryos at day 3. Am I right? Okay.
Yes.
What if a
So
woman
in
gets
fact,
spontaneously pregnant? What do we do?
Yes. There are a lot of spontaneous pregnancies even reported. And we cannot stop people from reproducing, or we cannot tell them that, no, you cannot have babies now. It's not ethical and neither justifiable. And even that
Yes.
Dr. Padmarekha Jirge, we did analysis of her patient data, and we did find exactly the same thing. That a lot of patients are actually requesting IVF now because of the job conditions and the stability at home. So we cannot decline that, but that means it would require a lot of information providing to the patient,
Yes.
especially the risk involved in first trimester infections and the through pregnancy infections, because we know very little except for the evidence. There is some evidence for mother to child transmission. We don't know what are the long term sequelae of this. We don't know what all other tissues are expressing, are likely to get infected by the virus. And of course, we have now data saying that the first trimester pregnancies might be
Yes.
more susceptible to infections.
Yes.
So this information has to be passed on to the patients, and they have to be told very carefully that this is what it's a high risk business, and you need to be careful while being pregnant. And of course, as the WHO and ICMR recommend, that we have to have tell them the best hygienic practice, maintain maximum social distancing, reduce the visit to the clinics, avoid a lot of visiting a lot of places, avoid people coming over. So these kind of situations. But in a country like India, where many of these things are tough, it's quite challenging, and this is something
Very
that we need to
difficult,
start
absolutely.
developing.
The number of questions, doctor, is it right time to get pregnant now? Can I get pregnant? To be honest, we don't know the answer. We really don't know the answer. But yes, we tell them if you wait, it's better, because God forbid you pick up infection. We don't know what harm it can cause. Because of the experience we've had with the Zika virus, it's known that there are so many problems related to it. You wanted to discuss one case report, right?
Yes, so that was interesting, and this was in fact thanks to Rahul and the ESIS hospital, where when we started this registry, and slowly as people started coming to know that there is a possibility of this virus getting transmitted, there was this case which they came across where the woman was infected when she was around 8 weeks pregnant. She was asymptomatic. She had absolutely no reason to get tested, but because she came in touch with some positive patient, she got herself tested. She was asymptomatic, and absolutely nothing. And by 13 weeks, she came home for the first ultrasound, and there was fetal hydrops there. And the fetal cardiac activity was absent. And then frantically, Rahul gets a call from them that, look, this has happened, and what can we do about it? So we thought that, okay, let's see if this is an opportunity for us to see whether the virus has transmitted from the mother to the baby, and is that a cause of fetal hydrops. And that's where we got in touch with them. We got the placenta samples of the first trimester because she underwent, obviously, she underwent abortion. And then the amniotic fluid was also collected, and we started seeing that is there a virus there. And we did a lot of experiments to go through that. So as Madam said, that it's a collaborative effort where the clinicians, radiologists, we as a laboratory people came in together and we started putting our minds there, and we found something quite interesting. I'd like to share a slide here, but I want to show that there is real virus there in the placental villi. First time it's right, of this woman. And you can see my screen, right?
It's opening. Yeah, we can see it now.
So this is the first trimester placental villus section, which was from a mother who was infected at 8 weeks. Remember, she was negative for the virus, but you can see all this lovely red color sitting inside, and these are the placental villi here. And you can see this virus there sitting and multiplying rapidly. These are the syncytiotrophoblasts. These are the syncytiotrophoblasts. These are the cytotrophoblasts. And you can see inside this red color is the antibody which is used to stain the presence of the virus, the S protein. And you can see happily the virus is multiplying in the placenta, and it's like tons and tons of it. Remember, she's negative by it. And after a lot of testing, so my colleagues in the institute tested the amniotic fluid for RNA of the virus, and even that was positive. And what we now think is that when the woman is pregnant and there's a virus in her lung, that the virus starts replicating. And so unlike Zika, this is not a blood-borne virus, this is a respiratory virus. 90% of the women might clear the virus in the lung and she may go well. But in a fraction of women, we think that the number is about 10%. That this virus can escape and that it can go in the bloodstream. That is called as the virus shedding or plasma shedding. And now it gets to the placenta. As I told you earlier from our work, that the placental syncytiotrophoblasts, the cytotrophoblasts, and the fetal capillaries, they have the ACE receptor. So from the mother's blood, it reaches the placenta. In the placenta, it gets inside the placental cells, and it can happily replicate there. So the amount of virus doubles, triples, quadruples, and it increases in number. Remember, the maternal immune system at this time is very low. It doesn't like to be it is not activated, so the virus is not detected by the maternal immune system in the placenta. It happily gets replicated, and then from the mother, it can get into the amniotic fluid and then the baby. So we think that like the Zika, through a respiratory tract, the virus can get infected, and in this case, probably the same thing happened. We could not test the baby here, but we know that the babies, the first trimester, right from the first trimester, the same ACE receptors are there in the fetal liver, it's there in the fetal erythrocytes, they're there in the fetal lung, and then the fetus can also get infected and can have long-term sequelae to it. So this is what we think here, that the virus is most likely going to infect if the virus infects the mother, it can then go ahead and have a vertical transmission, and this can have a drastic consequence.
So even though it's not a blood-borne virus, it can still transgress the placenta, which is actually facilitating its multiplication and allowing it to reach the baby. It's something like that. So actually, Dr. Rahul, because you're having the registry, what kind of problems you have been seeing in those 3,000 patients who have come to you? I mean, are you seeing that there are a lot of stillbirths happening, or a lot of placental abruption, or is there a lot of preeclampsia? Because it is known to affect even the vascular system. So in short, what kind of complications you are seeing in those patients? Do you have that data?
Yeah. So actually, we are analyzing the data of the registry, but we did carry out the interim analysis of the single center data of the NIRRH hospital, where my collaborator, Dr. Neeraj Mahajan, he actually manages the COVID-19 pregnant mothers. So what we found that in 700 women with COVID-19, we analyzed this data and we found that there is a high proportion of early fetal demise in first trimester. So there is a difference between the abortions in the first trimester and the second trimester. We also found that the stillbirths are high in these women, and we compared the pre-pandemic data with this pandemic data, and we found that the rates of stillbirths are high. We also found that the pre-eclampsia is high in these women with COVID-19. So, and other pregnancy complications are also being reported. So we are actually going in depth into the data, and we are trying to compare with the other 18 medical colleges. So I think end of the by end of the year, we may have the complete data of these 3,000 women with COVID-19.
But you're
And
seeing more of mid-trimester problems or full-term problems?
so when the pandemic started, we had mainly the third trimester women who were approached for the delivery. But now we have the data from the the second trimester also, and the first trimester also. Okay. I would like to highlight here that we had some interesting observations from these pre-COVID registry data. We had 3 cases of postpartum psychosis. We had found the co-infections of malaria and dengue also.
Yes. I read your papers on that malaria, dengue, and psychosis, and I was wondering whether psychosis, which is there, is there any evidence to show that the virus is affecting the neurological system, or is it because Indian women are so used to having so many people around them when they deliver? There's so much of support that when they have COVID positive, they are alone by themselves wherever they are. And could that be the cause of psychosis, or could it be that there is evidence to show that there are neurological or neuropsychiatric disturbances in these women?
So when we started this registry and when we started analyzing the data of pregnant women with COVID-19, we had this small observation that majority of the women with COVID-19 who are coming for delivery had anxiety. And like a lot of information was passing through WhatsApp and other social media platforms. So they were really worried. With respect to the association of the SARS-CoV-2 in postpartum psychosis, I think at this moment we can't say that it's because of the SARS-CoV-2 infection, because we'll have to do the further studies. But there is evidence in the literature that there is a presence of SARS-CoV-2 in the central nervous system. And we also found that there is one paper which says that the psychological symptoms or the psychiatric symptoms such as delirium could be the presenting symptom of COVID-19 in asymptomatic patients. So we also had one case of delirium, and which is under revision now in Asian Journal of Psychiatry. So in these particular 3 cases, what we can say that we found the coexistence of COVID-19 in these women, where we found the postpartum psychosis. The important point here I would like to stress is that, and we all know that around 35% of these women with postpartum psychosis, they pose risk to their infant, and they also do the same. So it is essential to look for these warning signals, identify the psychiatric symptoms at an early stage, so that these women can be managed appropriately by the psychiatrist.
For counseling.
And for counseling also.
Yeah.
I'll interject here and say that the ACE receptors for the virus are present in the central nervous system, they're present in the brain, and the virus can breach the blood-brain barrier because the endothelium can get infected. So there is both psychology.
They have also found persistent virus in the GI tract, like in the stool. It's persistently there, and that could be another source of infection for a long time. So there are, sort of so much going on that, honestly, we don't know finally what's going to come out of it. It seems to be affecting every part of the body. Like Dr. Rahul said that malaria and dengue is occurring more commonly in those patients who have COVID. As a matter of fact, recently one of our staff had fever and she was diagnosed by her GP as malaria, treated for malaria. She called, she says, "Can I come to work?" I said, "First do the test, let's see what you have." She says, "No, I don't have anything." I said, "Please do the test." She did the antigen test, negative. I said, "No, I want an RT-PCR." She did an RT-PCR, positive. So she's quarantining at home and she will come now after when she's notified. So in short, there are so many areas. Dr. Smita, I have a question for you. What I want to know from you is that what would be your advice as a director of the institute, keeping in mind that I think Rahul's papers talks about 1 out of every 9 women only is symptomatic. 9 women are asymptomatic, 1 is symptomatic, according to the data which you have published. So question is, what do we do in terms of public health policy? How do we do it? People are scared to do the test because when they do the test, if they are positive, they are quarantined at home. They lose work. They cannot continue working. They are put inside one room. Everybody doesn't have a space to quarantine themselves. They don't want to go to the government hospital. So it's a nightmare actually just now. So what would you recommend we do? Leave them alone. Don't do the test. Let them
No,
work.
no.
What do we
Uh,
do?
I think when ICMR and Government of India were trying to develop these guidelines and strategies of testing, way
Yeah.
back in April, they took a call that all pregnant women,
Yeah.
it is called a universal screening of pregnant
Correct.
women.
Correct.
So it is mandatory and it is good for them. And in that particular paper, we are trying to say that how the government policy
Yeah.
helped us to detect these asymptomatic cases. And we
Hmm.
had out of almost like 10% were symptomatic and 90% were asymptomatic. Suppose
Hmm.
this particular strategy was not there in place, we would have got more infections transmitted to these women, not only in
Yeah.
the community, also to the healthcare workers.
Of
So
course.
as of now, it is mandatory and compulsory, and it is good for the pregnant women itself and for their family members. So I would recommend that each and every person they should get this test done,
Yeah. As a
and
matter of fact,
the
it's
care has to
mandatory.
be taken.
Yeah, it's mandatory when you travel by air, you go from one place to the other, it's mandatory. You get into the hospital, it's mandatory. You get surgery done, it's mandatory.
Yes,
You
yes,
do IVF, it's mandatory. So everyone is actually being, compelled to do the test. And I think it's necessary to do it because of the complete asymptomatic nature in a large percentage of women. So, in
correct.
short, there are two last questions I want to ask. I don't know who will answer. The first one will be, will there be any role for artificial intelligence in this COVID lab? Collecting information, putting it into the computer, and putting in a large amount of data and coming up with something which is good, because we are having so many therapies which we are using. One day it's chloroquine, one day it's something else, today it is remdesivir. So, today there's plasma therapy. So, if everyone in the world puts it together into an artificial intelligence system, I think we'll have good answers coming into this.
Now, already I can take up this question. ICMR
Yes.
has initiated, just
Ah,
like how we have this pregnancy COVID registry, ICMR has already initiated a registry for general patients.
okay.
So, again, it is open for all these hospitals. Some of the hospitals are already partners. Huge data will be captured, and eventually it will get through the artificial intelligence mechanism. It will come in a big way because it will give us some kind of algorithms.
Yes,
What kind of like clinical features, then what kind of treatment was given. So it will tell us,
of
it
course.
will differentiate between the mild, moderate, and severe. So who got which kind of treatment and how effective it was, how long did they stay in the hospital, how long
Hmm.
did they take to clear off the virus. So all those
Absolutely.
questions will be looked at, and definitely it will play a major role.
Yeah, I think it is necessary because today there is so much of post infection morbidity which is happening. And
Yes,
it's not only, suffering from the infection and getting over with it affects different organs. And a lot of morbidity is being created because of it. So there's a deep concern on that too. So, my last question for the day, of course, I've kept it for the PCOS patient. So, I would like you to reply to this, Deepak. what about PCO patients? The reason we took this up through the PCOS Society because we believe that PCO patients are more at risk for this problem as compared to the non-PCOS. So can you shed some light on that?
Yeah, sure. One of the things that is always questionable is, are the gametes likely to get infected? And there is now data to show that yes, the ACE receptors are there in the ovary, but at least from the two patients, this paper was published in Human Reproduction recently, where there are two patients in whom ovum pickup was done when the patients were positive. So this was inadvertently done without realizing, because the tests were
Without
not
realizing.
then. And the good news there is that the oocyte does not seem to get infected by the virus. But we don't have any data, remember, on the follicular fluid. We don't have any data on the granulosa cells. So at least the oocyte does not get infected. There is at least some evidence towards that. So which is nice, and which also I believe
Two patients.
two patients, yes.
What
But at
about the
least because
sperm?
yes, the testis also is likely to get infected. So in course of active infection, if there is active infection, there is a very high chance that the testis can get infected. Orchitis is one of the symptoms there, and the seminal plasma is also positive for the virus. So during the course of active infection, yes, there is chance that the father can transmit the infection. Also, there was a recent paper in American Journal of Obsgyny where they found that at least in 7 to 10% of women, even in vaginal secretions, the virus can be detected during the active course, whether they're symptomatic or asymptomatic. So the virus is probably found in almost all body fluids. So of course, one has to be careful while doing ETs, while preparing the gametes, and so on and so forth. Now, getting to so the ovary does not seem to be spared from the virus, but we don't have any data to say that it is safe from the virus. The
No,
machinery is there.
the question is on PCO patients, whether they are
Yes.
at a
So
higher risk because of their metabolic issues, their insulin
yes.
resistance. Are they
Yes.
more at risk?
Yeah, so definitely, because we know from literature that men are more likely to get infected with the virus than women, and it is because of androgens. And PCOS women are hyperandrogenic. Diabetics are more likely to get infection, and PCOS, a proportion of PCOS women are diabetic. Obesity is a risk factor for COVID-19. PCO women are obese. And inflammation is one of the high risk factor for COVID-19, and PCO women suffer from inflammation. So
Chronic
the most
inflammation.
like there's chronic inflammation there. So in all possibility, there is a good chance that they might be infected by the virus, but currently there is absolutely no data. So I think it's time again. Time and again, I keep saying that as researchers, as clinicians, as societies, we should all get together and
I
talk
think
about
as a PCOS society, we may come to you to work on this aspect
it.
and
Yes.
look at how we can take it further with PCO patients. And I thank all of you tremendously for bringing up so much of knowledge and so much information. And we've had a great, at least I've enjoyed myself. I don't know if it was good for you, but I'm sure there'll be a lot of questions coming in from the audience. Unfortunately, I didn't get time to take their questions because I was busy taking mine. And therefore, what I'm going to do is whatever questions we receive, if you don't mind, I will email them to you so that we could work up a way where we could respond to those questions. If you all agree, I can do that.
Definitely, ma'am.
Yes.
Definitely. We
Good.
can do that.
So
And it
thank you
was
so much, everybody. Dr. Smita, thank you very
Yeah,
much
thank you.
for,
It was very nice.
yeah, thank you. Thank you so much for being with
Thank
us.
you
it was
so
And
much.
absolute
thank you,
pleasure
Deepak,
being
and
here.
thank you, Rahul.
Thank you.
And I thank the organizer, the platform Doc Mode, for supporting this event. And thank you very much, all of you. Bye-bye. Good
Thank you.
night.
Thank you.